Yeah, we've got a beautiful glass sponge there on a stalk.
Snap zoom there, Jonathan, please.
Probably,
Ooh,
That's great.
very cool.
Steve said it might be a Farrea, F-A-R-R-E-A.
All right, there, Jonathan. Full wide, please.
There's another rockfish.
All right, coming up.
Coming up. The sea urchins we're seeing very commonly here are quite large. They're about 20 centimeters across. If you were watching earlier, we collected one, and you could see how they're very unlike sea urchins you might be used to. They have quite a soft body, which we call a test. Its skeleton is very flexible in terms of the plates that make it up. It does have a lot of spines, and it also has a lot of little things called tube feet with which it moves around. These tube feet are what all the echinoderms have, so sea urchins are related to sea cucumbers, and then also the sea stars, brittle stars, and the crinoids. Sea cucumbers and sea urchins are each other's closest relatives, and in the case of sea cucumbers, they've lost all of those plates for the most part and just reduced to some little calcareous elements in their body wall. But quite a few deep sea urchins like this one we're seeing here, Asthenosoma, are also very much reduced in their body plates. But the Asthenosoma can defend itself. It, like some other sea urchins, have some little jawed projections coming out of their body that are able to bite effectively and envenomate, actually. Anyone that might come along that attempts to harass them. There are some even in shallow waters that can give you a very nasty envenomation and a very painful sting. So when this Asthenosoma comes up tonight, we'll be handling it very carefully. We've collected this one before from Costa Rica, and I'll be quite interested to see if it's the same one or it's thousands of kilometers to the south, whether this turns out to be a deep sea species with a long, large range, or whether we might have a case of a different species here off California. Just passed by a large sea anemone. We're now going over, we're starting to see more of these large rocks. We're hoping to come to a wall in the latter part of the dive.
Is that one of those Venus flytrap anemones?
Yeah, Venus flytrap, and then also pom-pom anemones.
Ooh, really cool
Snap
colors
zoom
there.
there, Jonathan.
Gorgeous.
That's great.
Wow. So delicate. Right next to each other.
Okay, full wide there, please.
I feel so lucky to see these. Nobody's probably ever seen these before or will see them in the future. Happy Halloween, everybody.
Happy Halloween.
Oh, yeah.
What better way to celebrate Halloween than dive the mysterious depths of the deep sea? And I have a little Halloween deep sea joke for you guys.
Yes, please.
Where do mummies like to go swimming?
Is it the Dead Sea?
Yes.
Wow.
That's great.
Does anybody else have any good jokes?
Trying to conjure one up now in my brain.
Yeah.
Not working too well.
I have a iPhone note list of bad
Deep
jokes.
sea jokes?
Deep sea jokes,
well, it's
right.
mostly
Just
like
dad jokes?
jokes that have come up on watch, Yeah.
Oh, very cool.
They're usually very hyper contextual and not funny. Unless it's 4 in the morning after a long watch.
Yeah, snap zoom there, Jonathan, please.
Ooh,
Beautiful sponge.
they like radial.
Some little inhabitants kind of nearby on the sponge.
So Kyla, people were laughing so much that the scientists on shore didn't hear the answer. Can you give the answer to your joke?
Oh yeah, the Dead Sea. The Dead Sea. Where do mummies like to
Boy,
go
Jonathan.
swimming? And the answer is the Dead Sea. And I have another one.
Oh, yes, yes,
Yes.
yes. This is going to be comedy hour. Love it.
What do vampires cross the sea in?
In?
Often shit.
Blood vessels.
Oh, that's good.
That's pretty good. These are great.
Good one.
Keep them rolling.
Thanks, Google.
So zooming back a little bit, we've got an ID on that stalked sponge that we saw. And it turns out it's a Sacocalix, which is in the Euplettidae family. So phoriids are typically bushy and typically not stalk. So if we see one of those again, we'll have to make a note. A little bit different from the phoria white sponges that we've been seeing. Thanks, Steve.
Curious little rocky outcrops, all these.
Another one of those anemones.
It's called liponema, if you're logging that, Kyla.
Got it. Thank you.
Liponema.
The pittings here in the sediment are quite curious.
Yeah, and the different mounds of sediment.
Yeah.
Are those bioherms? Or are they geological?
Not sure if they'd be biofilms. Paul, do you have any thoughts if these would be biofilms on top of any sediments?
Oh, bioherm.
Oh,
Bioherm.
I see lots of little pores in the sediment too.
yeah. So those could be.
I think those are anemones with their tentacles sticking out.
You want to see?
Just kind of little
Could
red
zoom in
stalks.
on one. There's
Yeah, definitely.
Yeah.
Go ahead, Jonathan, and push on in.
Oh, yeah.
We'll just
Whoa.
Very
maybe I'll
cool.
sit down for this.
They're not pores, they're anemones.
Ooh.
They're so cute and tiny.
Ah, before the dust cloud gets there. Okay, we're looking up here.
I wonder why they're also spread apart.
Ooh.
Ooh,
Sorry, little.
Startled.
shrimpus.
All right, go ahead and push in on that one there,
It's all the way.
Reg. Sorry,
There's-
guys, for that.
there's a
Go ahead.
group of anemones. Well, they're not really anemones. They're called tube anemones, and they have a very distinctive sandy tube, and then 2 rows of tentacles. And we have a lot off the California coast in shallow water. I was just trying to see if these were tube anemones, cereanthids. but I think they're actually true anemones, and there are some anemones that are burrowing anemones that will live in sediment like this. Probably a bit of a challenge. We'd have to push call one to catch one. We're trying to get up the slope, so we probably won't. Some of the other burrows we're seeing, we can see tracks coming out of the burrows with clean
Push in there
sand.
quick there, Jonathan. Great.
So those are probably a kind of worm.
Yeah.
Yeah, there's a nice view of one of those. This is a real anemone.
Tentacles flying in the...
If we disturbed it.
Go wide there, Jonathan.
You want to take one? Do we have push calls left?
We do have two push cores left.
Yeah.
So the issue is we start at the bottom a little. If we're going to take another push core, ideally we would come a little off the bottom, trip at Niskin, and then come settle down and take a push core, and
stop the ship.
we could do it right on top of that anemone. Then we get a
Do you want to
little
do it
bonus.
here, or do you want to wait for a clearer spot?
Coming up.
Whatever you're comfortable with, but
They seem to be dotting the floor here.
Okay, we can stop the ship. Bridge now.
Yeah, maybe the next time we're in a clear area without rocks nearby.
Hold position. Do we see any?
A little red jelly. Another one.
Science, we're just gonna kind of get ahead of Argus while it swings. You can kind of see us starting to swing into a feature. A little bit. And we'll see if we can find those wherever we settle out to.
Can we get a zoom there?
Is that an
an octopus?
octopus?
Yep.
That's an octopus.
Looks like it.
Got a Halloween treat.
Yes.
Oh my goodness.
Yes.
Oh.
In the rocks there.
Go ahead, Jonathan, and do a snap there.
Oh, look at that.
Wow.
Oh my
cool.
goodness.
Look at that texture.
Very cool. That's
Big
incredible.
eyes.
Almost looks like she's finessing.
Spooky.
Also really cute.
Noah says
Also
it's
cute.
National
Cool
Octopus
white.
Day.
Is it National Octopus Day?
How appropriate.
It is
There
now.
we go. And Halloween. Very cool. Thanks for that zoom.
Sure. Sure thing.
Was that our first cephalopod?
I think it was.
I think
of this
so.
expedition, yeah.
Really cool
pretty cool.
feature here.
Yeah, this is awesome.
Flow.
Wow.
The sense of the scale on Argus is really cool.
That's true.
All right, I think Argus is pretty stable.
You mind if I zoom in with Argus?
Yeah, go ahead. Watch the sonar. All right, Paul and Greg, you want us to go back and see if we can get one of those burrowing anemones? Or
Oh, no, thanks, Jessica. Let's keep going.
Roger.
Hopefully we'll hit another patch of sediment. It's not super urgent.
Okay. Roger that. Thank you.
We'd love to get as far up the slope as we can.
Raj. I'll just get ahead of you
We've
there.
crossed 1,000 meters now. We're at 987 meters. We're definitely seeing a lot more rock. It's getting a bit steeper.
Greg, correct me if I'm wrong, but it seems like most deep sea anemones are solitary, whereas nearshore are colonial. That's just my observation.
Look at that feature. Oh my god.
Oh,
So gorgeous.
Very steep.
most anemones are solitary. So there's a few inshore. I guess it's a bit like Steve was explaining. We had a talk from another scientist on board about the word coral, and it's applied to many different groups that have evolved independently, and they're called corals. And the word anemone can be applied to various different independent groups. Like I called something a tube anemone before, but it's actually a completely different group to a normal sea anemone you might see in a tide pool on the seashore, or these pom-pom anemones, or the Venus flytrap anemone. But there are other things that are colonial that we also call anemones. Off California, we have the beautiful jewel anemones that are colonial. There's a seaweed whip, or no, that's another one of those Sacco-calyx that Steve told us about, a stalk glass sponge.
family. That's beautiful coming off of there.
We're just out of reach on our tether, but we can admire it from afar.
That's a nice
Unless,
shot.
Jake, you want to come down for about a few meters there?
Yeah, I can come down a few meters.
Thanks for that anemone explanation. Super interesting.
So
Jonathan, go ahead and do a partial there, please.
I assume the stalked adaptation is to get up off the bottom into more current?
Yeah,
You can push in a
get
little
away
more.
from
So, so you see that with some echinoderms too in the deep sea, right?
That's beautiful.
So we haven't collected this one yet.
we have not.
Do we have a good...
You want it?
Wow.
It's pretty big.
Jessica, is it feasible to get down there and make a collection of that sponge, or?
Let's see, full wide there, Jonathan.
It's I'm not sure where we'd put it, Paul.
It is
We
quite
could put
large.
it in the forward box. It's about 50 centimeters long, so it's with the stalk.
It'll likely break up too, potentially. Would it fit in that forward box, Jessica, you think? The inboard forward box?
Yeah, we can also take a piece of it, or a big piece of it.
Yeah.
Yeah, maybe a piece.
Yeah.
And then it can continue on.
That's gonna be an interesting grab. Yeah, we can try it out.
I think with the swing, I'll stop here.
Raj.
And it'll come towards you.
Roger that. Thanks, Ernie. Maybe a full wide there, Jonathan, please.
Maybe a little more.
Paul, are there any glass sponges that are known to be chemically defended, or do they really rely on their spicules?
Yeah, they're not well known for chemical defense, but seems to be the majority of what we see down here. So
yeah.
Has
anyone looked at their microbiota, their microbiome?
Not that I'm aware of.
Yeah, could be interesting.
Yeah.
All right, Rennie, you want to do me a favor there?
Ready?
Yeah.
So this sponge is on long glass spicules, rods that extend all the way to glue it to the rock. And some of these spicules will end up being a single long rod.
Can you go in there tight, please, Jonathan?
Extraordinary in that they've been secreted by a single cell or group of cells. And then it's coming up to a big chamber, and the water comes in through the outside, and then it's being pumped out through the top, where you can see a series of holes.
Wow.
And there may even be some creatures living on the inside of this sponge when we open it up, which have a safe place to live and probably are able to even source some food from the sponge. This particular group of sponges is known, another group of them are called Venus's flower basket. euplectella, and that's well known for having a pair of shrimps living inside it, encased by the meshy glass network.
That's great.
And they can't get out. They're stuck in there for life.
Jonathan, are you full wide on Argus?
And these sponges are given dried as a wedding gift in Japan for a metaphor about the state of matrimony.
Of being trapped.
Trapped in a beautiful place.
Science,
One way or the other.
What kind of a big hand grab
look
is
on the
what you're
bright
looking
side.
for there,
Yes,
Paul?
please, Jessica. That'd be great.
Okay. Actually, I'm going to get a little lower in there. Oh, that's not what you want.
Looks a lot less brittle than the Phorea.
That's for sure.
Quite strong.
It's tearing.
It is, yeah.
Do we know how old this sponge is?
I'm not aware, Kyla, how old these sponges live. Given the low amount of nutrients in the deep sea, and I assume producing these spicules is energetically demanding to make this silica glass rods. It could have taken this animal quite a while to grow this big. Hopefully it'll be able to repair the damage we're doing to it reasonably quickly. It'll still be able to feed. In a way, sponges are
I'm going to take that
unusual for animals. They don't have a nervous system or muscles.
Oh, hi there, Jonathan.
They're kind of an association of cells. So it wasn't hurt in any way when we just sampled it. It's certainly disrupting it.
Actually, do you guys know if that anemone will be floaty? Sorry, anemone sea urchin is floaty?
It'll be a little floaty, but it should be okay.
Roger that.
Because it got punctured, I think it lost most of its fluid, so it should just be sitting on the bottom.
Roger that. All right, go ahead and open up the tool tray there, Jake.
Roger. Just confirming this is going to be sample NA124 071.
071.
And Paul, would you like 2 handfuls since this one was relatively small?
Let's see how this goes. I'm a little worried how floaty this is going to be.
Roger that. So I'm pushing myself up the
Yeah, it's going to be challenging. Close it quick, 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 quick.
Looks
like it's staying.
Pretty good piece.
Yeah, I'm happy with that. If you can close it and we can keep it in there, then.
Have to put
Perfect.
put something on top of it.
Okay, so you're happy with that size of sample?
Yes, thank you.
Thank you. And thank you, Sacco Calyx.
All right.
Kyla pulls added more, Steve's added more about the things that are called anemones and not anemones, and what a true anemone is called actinaria. But here he's talking about the things like the jewel anemones, they're called corallimorphs. If you remember his tree from last night.
All right, ready.
Ready?
Ready.
Bridge nav.
Pretty ribbon eel or some sort.
So Greg mentioned the sponge microbiome, and sponges are particularly interesting because, for one, they feed on bacteria that they filter out of the water, so they inherently have bacterial communities associated with them that they filtered out of the seawater. But some groups of sponges are well known to also maintain dense populations of symbiotic bacteria within their mesophyll, sort of the middle section of the sponge. And in terms of thinking about natural products and biopharmaceutical potential, there have been a number of examples where people have collected sponges and found interesting molecules, and later been able to determine that it's actually the microbes that are living as symbionts within the sponges that are the true source of those molecules. So all of those examples are from shallow water sponges. So I would venture to bet that nothing's ever been done in terms of exploring the microbiomes associated with any of these deep water sponges. So I suspect that we're going to be doing a little bit of that.
Not seeing any corals down here, are we?
No, nothing yet. Got some really interesting rock formation. Still kind of climbing that wall. Really cool shot from Argus.
Hi, this is Devin in the lab. Can you tell us the depth we took the push cores at?
The depth that we took the push cores. Sure. So the depth of those, just give me 1 second, I'm going back through our sample notes.
One two nine six
Assuming-
meters.
yes.
1296 meters.
1296, thank you.
So we're passing through about 960 meters now. We've seen from the beginning of the dive at 1200 meters, or nearly 1300 meters, we've come up a few hundred meters. Our goal is to get up the eastern side of what's called Northeast Bank to near the top of it where it plateaus. It once was an island, and we're expecting, if we can get that high, we'll see a lot of corals and sponges in the higher oxygen area. We've got quite a ways to go to get there, and we will be collecting rocks along the way. We're in Southern California. We're offshore from basically Los Angeles is the nearest big city. Magnificent pom-pom anemone, leponema there, rockfish, sebastes, a couple of prawns, or shrimps, we're not sure what they are. We're interested in the rocks here that are encrusted with today's dive, probably ferromanganese, potentially phosphorites. We'll see when we bring the rocks up, and our geologist on board, Kira Mazel, will tell us what we have. And we'll also be sampling the various kinds of animals along the way. So far we've picked up a nice, beautiful sponge. We've taken some push cores, collected and lost a sea cucumber, which was very, very buoyant, or neutrally buoyant, and it came out in the wash when we opened the box. And also one of these sea urchins that you can see in the frame now. A lot of sediment on this slope, so we'll be looking for some areas with maybe some more exposed rocks that are easy to pick up. We need to collect a few more rocks along the path we're going. Could we start a transect, please?
Can indeed. You good with that,
Sure
Jess?
thing. Yep, let's do it.
All right. Ship move is continuing. 305.
Raj.
This is going to be transect number 2.
Copy. Let's do a quick DVL reset before you're in audit, if you are going to audit. Okay.
All right, ready for you.
Good to go.
And just to confirm, have we started the transect?
Yes.
Yes.
Beautiful.
Just started.
Thank you.
Just started.
All sorts of holes and pock marks in this rock. How it's been weathered like that, not immediately obvious to us, but we're biologists, so we don't have a geologist on watch with us this afternoon to tell us what's really happening.
Or those beautiful anemones.
It's quite a ridge.
Mm.
Haven't really seen a formation like that yet.
Or it's just one big giant rock?
Lava tube, maybe?
Hagfish on the left. Kind of
Swimming the
eel.
eel.
Yeah.
Very cool.
That was very ghoulish. So if you had to choose, would you say that either a vampire squid or a dragonfish is most representative of Halloween?
Vampire squid.
Yeah, vampire squid, but we're biased because we saw one. Couple, actually.
I agree. Vampire squid.
I think the dragonfish with the really big teeth, though.
Right.
Pretty impressive.
That is true.
And those shimmery, dark scales.
Yeah, a toothy smile.
What about that like gulper eel?
Oh yeah, gulper.
Big
Yeah.
jaws on it.
Another good contestant.
The poor vampire squid gets a bad rap. Its Latin name is Vampiroteuthis infernalis, which means vampire squid from hell.
Oh, no.
But it doesn't prey on anything particularly. It sits in the water column and it actually is a detritivore. It eats marine snow.
Oh, poor
So
thing.
it's hardly a terrifying creature at all. Although much unlike its looks, it's eating the marine snow falling to the seafloor.
Totally misunderstood.
I have misjudged the vampire squid. My apologies. My vote goes to the gulper eel now.
I think anglerfish are pretty spooky. The deep sea female anglerfish with her little
Yeah.
dwarf male attached.
Right. Yeah. Talk about sexual dimorphism. The male anglerfish is basically a parasite.
Well, we don't want to get into a philosophical discussion about the utility of males, but we could.
I think they're recently, if I remember right, my inbox from Science or Nature, that they're being studied for possible whatever compounds allow the female to not reject the male when the male kind of bonds with it, in looking at organ donors in humans or organ donation in humans. And
Yeah, the male completely fuses with the female and her circulatory system then unites with his to keep him alive. So there's definitely an interesting immunological story there.
That's insane. That's crazy. Evolution is wild.
And there's a lot of chemistry involved with these associations. For example, there are some crabs that the males can tell when the females are molting because they sense a chemical cue, and they mate when the females are molting.
Do you guys mind if I change my heading on this transect to face the rock?
No problem.
Sure.
Thank you.
Thank you.
So the male can sense through a chemical cue when the female's molting, but the problem is these crabs are also carnivorous, and I should say they're cannibalistic, and so the male's also tempted to eat the female.
Oh my gosh.
So the female emits a chemical cue that suppresses the male's feeding stimulus.
What?
So there's these back and forth chemical cues going on to help ensure successful breeding.
Wow. That is an awful first date. Interesting.
That's fascinating.
Yeah, the male basically kidnaps the molting female. I think they can sense before the shells actually start to get soft, and they basically hold them prisoner until their shells soften. And then they release them after the shells start to harden again.
Sounds like the female's in charge.
might be a bit of a pause here, Jess.
Raj.
Sorry about that.
No, no worries.
those chemicals be considered pheromones?
Exactly, correct, yeah. This is something that most people don't appreciate in the ocean, is that most communication is chemical, right? And especially down in the deep sea where there's not a lot of, not relying very much on vision. Of course, there's some bioluminescence and things that organisms take advantage of, but just like when yesterday Greg set down some colonization experiments, and some of those were bones that had a little bit of meat left on them, and crabs were coming from all over the place. And so they can pick up these scents very far away. I mean, obviously, food's not widely abundant down here, and so it's pretty critical to be able to detect a food source, even from long distances. So very acute sense of chemoreception. And that includes mate recognition, that includes predator avoidance, predator detection, chemical cues govern where larvae settle with invertebrates that are looking for just the right kind of surface to attach. It's the chemistry that's really guiding many of these behaviors that go unappreciated. And very hard to study too, because a lot of these molecules are water soluble, and they're produced in very, very low concentrations that these organisms can sense, but it's very hard to isolate them and identify them because they're in small, low concentrations, and they're often very small molecules. And in many times you need a bioassay to identify them, right? So if you want to know if it's a molecule is a chemical cue, and it, say, attracts an organism's larvae to settle on a certain substrate, you need to develop an assay to test that, and that's hugely challenging in itself. And then figure out which molecule is actually responsible for the attraction. It's very complicated. Small sponge there.
Thanks, Paul. That was a great summary of chemistry, chemical biology in the sea. And if you ever come and do a course at UCSD, Paul does a great course for undergraduates and graduates in chemical biology.
So there's one of those mushroom corals there for you guys.
Ooh,
yeah, right to the left there.
We can pause the transect.
Sure.
We might not get to see one again, if we can stop it.
How far in the transect had we come already?
75 meters.
Thank you.
Well spotted. Hopefully that'll hold
Pull
down
right
the
there.
sponge in the biobox.
Yeah. Yeah, hopefully when it retracts, it'll act as a little bit of an anchor. Might be floaty though.
I spent some time looking last night, and I couldn't see any records of any natural products reported.
All right, Rennie, you want to, oh, never mind, stand by.
These mushroom corals.
Thought I had it.
They might not have been studied before.
That's what I'm hoping.
Definitely have collected a few of them, but probably not with the same purpose as what you're doing back there. Just mainly for archiving, stuff like that.
All right, Rennie, go ahead.
So this is a colony. It's an octocoral. If we zoom in, we'll see the polyps coming out. Each have 8 tentacles. You might be familiar with another group that are called mushroom corals, which are solitary, and they're stony corals, true corals. And they are free, not really attached to the seafloor. But here we've got this colony of octocorals that have also been given this common name, mushroom coral.
Go ahead. Thank you.
And when we disturb this animal, probably the polyps will pull in and we'll be left with a ball. And the name of this was...
Do it fast. All right, Jonathan, go ahead and push on in there, please.
previously in the genus Anthomastus, but
Oh, that's
now it's
beautiful.
called Heteropolypus.
Heteropolypus.
Yeah, I can't get used to that switch.
Oh, that's
Only this year
beautiful.
did I hear about it.
Getting some gorgeous screen grabs. Thank you.
No worries. All right, there, Jonathan, full wide, please.
Lots of surface area.
I have a feeling that when we open up that box, I'll get pushed off the cliff. So we'll stand by.
Okay. Yep, just let me know.
Thanks, Jake. All right, Jonathan, go ahead and push on in a bit, please. Great. So you want all of it? Is that?
Yes, please.
All right.
You could take the whatever you can grab there.
Oh.
Even the sponge part would be useful.
Roger that.
Now
Might be
it's
stuck on the
retracting.
I think
sponge.
he is stuck on the sponge, yeah.
That would be
Very
a
cool.
good twofer.
It's like 2 types of sponges there, one growing on a dead one.
Got a little more zoom.
3 for.
There go the tentacles drawing.
Yeah, and it
polyps.
goes, like, I don't,
He's had a bad day. Oh, hey,
nice.
buddy.
That was like
Perfect.
a dog toy.
Are there 1 or 2? Let's see here.
Cool. There you go.
All right, well, at least we didn't squeeze him like a dog toy. Oh, I had there, Jonathan.
This is going to be sample NA124.072.
072.
All righty there, Jake. Go ahead and bump her out real slow there, please.
Inboard forward box.
That's great, thank
We're
you.
going to be putting it in the forward bio box, number 6, which is the white one. The one that we've been putting a bunch of different samples in. So hopefully this will hold down that floaty sponge.
Wait, this one won't float itself.
That is also true.
Nope, no floating.
Awesome.
Perfect.
Great sample, Jess.
Great.
As long as I can get my claw out of there without making a mess. All right.
Beautiful.
I'm going to close. There we go. All right, thanks, Jake.
Coming in.
All right.
All right, getting more relaxed. We're making some collections.
We still have a ways to climb, so we'll see what else we find.
Yeah, it'll be interesting. we've made, this is our third dive, and we've noticed that around 800 meters or a little below, really the biotic diversity picks up dramatically. So see if that happens again today.
Yeah, we're seeing a pretty dramatic decline in oxygen. And
Okay,
if we can
I'll
get up a
update
few
again.
hundred more meters, we'll
I think if
probably
you
start to see quite a
go
lot of other life.
something like 335, will get us ahead here.
Reg, did we want to still resume transect here?
Yes,
or-
please. Yeah, this
Yes,
looks like a good place to restart.
Reg. All right, I'll start moving ahead of you there.
Copy.
This is going to be a continuation of transect number 2.
It's a large sea cucumber there called Panicia. Has little walking legs underneath. A lot of these deep sea cucumbers are able to move around. As Paul was saying earlier before, these animals have to rely on smell, and even Panicia can detect better places to feed. So they'll accumulate around a thing like a whale fall, where there'll be a big explosion of bacteria in the sediment around the whale, and that's Panicia and other sea cucumbers that can walk can come from kilometers around and end up with large numbers, consuming all the bacteria in the sediment. Probably takes them a while to walk those kilometers, but they'll get there.
So that's a glass sponge that we've collected at least once.
Beautiful bright yellow color.
Sorry.
No, no
Just
worry.
nearly at the end, like 5 meters left.
I'll hold off here.
Mhm.
Do a quick gauge check.
Sure thing.
Here. Yep. Yeah.
Hmm.
Oh, I don't know who changed that.
Looks like Simrad. Color ramp the same as that or something similar. Mm-hmm.
All right,
Yeah, it might
start
happen
to
now.
move again.
Ooh, is that a crinoid?
Yeah.
Yeah, nice feather star there.
Beautiful.
Has 10 arms. It's not on a stalk.
Are we still looking for more stalked ones, Greg?
All right,
We
that's
see a
the end
stalked
of
one.
transect right there.
Okay, thanks,
Beautiful,
Renata.
thank you.
We'll continue upslope.
Roger. What else is on the wanting to collect list? So stalked crinoid and other types of sponges, or what's the?
Yeah. Not sure I might have the name here. I don't know if that's going to help, but
Some of the Gorgonians we
there
saw
was, yeah,
yesterday.
remember the Swiftia, the red branching
Mm-hmm.
Gorgonian, and when we started to see quite a bit of those, we also saw another sort of darker, bushier Gorgonian
Okay.
that we weren't able to manage to collect at the end of the dive.
And sea whip. We saw some sea whips yesterday we didn't get.
Whips.
Okay.
Did I see, I think I just glanced at the screen. I thought I saw someone collect something like a Swiftia yesterday. Was there something red and?
Yeah, we got a couple pieces of Swiftia yesterday.
Nice.
Nice red colors.
And Lisa's mentioned we want to collect some rocks, and so Lisa, we've left the fore aft, starboard aft bio boxes open for rocks. So our plan was to put an individual rock in each one of those, so
yeah,
keeping those spaces free for those collections.
Really haven't seen many free rocks. It's been very much a solid slope as we've been coming up.
Yeah, it's just some really interesting topography, but all giant slabs of rocks.
But as soon as we see some, Lisa, we'll be getting another couple of rocks. Now we're back on some sediment.
I know, we should think about a core if
we see the anemones.
Possible.
Oh, you want to get some more cores? Yeah.
I wouldn't mind.
This might be a chance to do
slope?
it.
Gonna try to stop here?
Look coreable.
Yeah, we can definitely try and see if it's coreable.
So before, let's pull a Niskin. Just trip a Niskin.
Raj.
If we could trip Niskin number 2, if possible.
All right, so theoretically I should be around 5 meters.
5 meters above the bottom?
Yeah, is that all right for you guys?
Yeah,
that's
that sounds
great. Yeah.
Great.
Thank you. This is going to be sample NA124
Copy that.
Niskin bottle number 2.
Don't we want to close now? There we go. Let's go this way. Apologies.